Good evening, everybody, and welcome to this episode of our Fireside Chats with me, Dr. Binita, Kane, and
myself, Helen Oakleigh, founding member of charity Long COVID Support and actor filmmaker, and passionate advocate to get to the bottom of what is going on with all the chronic conditions.
So as usual, the session's live and interactive, and we will be encouraging you to put your questions in the chat. Helen is going to hover backstage and keep an eye on the questions, and we'll be bringing our amazing guest for this evening on stage shortly. So yeah, please do remember that this is going to be recorded. It will be available on the YouTube channel for playback if you can't last for the whole hour. But Helen, for now, I'll pop you backstage and introduce our guest for this evening. Which is Professor Danny Altmann, who is a Professor of Immunology at Imperial College in London and also a trustee of our charity, our partner charity, Long COVID Support. Welcome, Danny.
Hi.
Genuinely really so thrilled to have you on, because you are an absolute giant in the long COVID world, and you do so much just in terms of the research, your advocacy and all the support. So thank you for coming on. It's lovely to see you. And we go back, don't we, to our Independence Age days, so that's how we know each other as well. Can we start just by getting you to tell us a little bit about yourself and then how you've got interested in COVID?
Yeah, so probably the best way to introduce it is to just say who on earth I am and what on earth professors of immunology do. So I've done it for many years. So I just love studying how the white blood cells in the body function in our immune system. And I started out with herpes viruses like Epstein-Barr virus, and then spent many years working in just different countries in the world on many different infectious diseases, viral and bacterial, including things like HIV and tuberculosis and all kinds of different stuff. And somewhere along the way, I also became an autoimmunity researcher, another disease of the immune system, looking at things like multiple sclerosis and rheumatoid arthritis and type 1 diabetes. So sort of big build up to the, when COVID-19 came along, end of 2019, beginning of 2020, it felt like something we'd been preparing for many, many years, really, for my whole life. And our lab at Hammersmith Hospital got very involved, working really 7 days a week. Because if you think back to those days, like early 2020, we were all terrified of this new virus, and it was a coronavirus. And the amount of stuff we really knew about it, about how it would behave, how good immunity would be to it, what it could do to you, how it could do it to you. You could have written on the back of a postage stamp. We knew almost nothing about it. So we all became really busy just doing stuff day and night, and publishing a lot of papers on it and talking to policymakers and government in different countries about it, doing a ton of stuff on COVID. And then, as this audience knows very well, by summer of 2020, it became clear that there was something really weird going on, and there were some people really not getting better and with persistent symptoms, and some of my best friends were among them. And we started having that discussion. And it was something really familiar to me, because part of our immediately pre-COVID world had been in Brazil on a virus infection that you get from mosquitoes called chikungunya virus, which is a bit like dengue or something. But the reason it's such a big deal there is that once you've had your initial fever and your acute infection from chikungunya, about a third of the people really don't get better for months or years and have a kind of arthritis-like condition that can really, put them out of action, put them out of the economy, put them out of their homes. So it's kind of familiar to me. So, long story short, we became long COVID researchers. And the only other, so I've given you a very long answer, but the only thing I'd add to that is through all that stuff, I became friendly with a long COVID activist who people might know called Jez Medinger, who asked if we could write a book together. Which we did, and that came out as the Penguin Long COVID Handbook. And since then, amongst other things, we've been quite an intensive long COVID research group recruiting cohorts, adult and pediatric, in the UK and all the way across Brazil, to try and understand what on earth is going on.
Yeah, no, I mean, there's so much to talk about just in your research, and we will get onto the Santa Fe conference, which obviously we you know, that's the title of this session today, is talking about all the research from all over the world. But I'm really interested, Danny, because you know the immune system, it's kind of the crux of everything really in terms of this condition, and you've done a lot of work studying adults and children. Are there any headlines from the research that your group has done that's kind of useful for people to know? Like what do we understand now that we didn't 5 years ago?
Yeah. So so understand might be too strong a word. We know a lot that we didn't know 5 years ago, and you know people often quote this fact, don't they, that if you look in the databases, there's something like 100,000 peer-reviewed publications now on long COVID. Which obviously isn't as many as it sounds, because they're sort of good, bad, and indifferent. But some of them are very, very good, and some of them are very, very important. So let's start off by saying why immunology is relevant and what we do. So from everything I know and everything I've ever done, I look at long COVID, and I think it's fair to badge it, amongst other things, as an immunological disease. Yeah, it looks like a lot of different immunological diseases. It's got elements in common with a lot of different inflammatory and autoimmune diseases. You know, that doesn't mean that I kind of negate or poo-poo other aspects like you know, coagulation aspects or other things, but you know, it's a complex body and all these things are interconnected. So I think it's a fair window to look through, is the window that I look through as an immunologist. And as immunologists, we have really busy labs full of really expensive kit that can do lots of things and measure lots of things. So if you start at the very high level, the immune system is all the kind of proteins, the soluble bits that float around in your blood, things like antibodies and cytokines and chemokines. And then it's all the different white blood cells that attack things and mediate immune responses. And I think during COVID, people became very educated in thinking about B cells, the cells that make antibodies, and T cells, the cells that mediate cellular immunity and kill viruses and kill tumors and things. But, you know, like everything you learn, that's kind of an oversimplification, because if you were going to, you know, immunology lectures and seminars, it's not two types of cells, it's many thousands or tens of thousands of types of cells that we can differentiate on our fancy cell sorters and things. So just by way of saying, it's a really kind of complex beast that's doing quite a nuanced job to protect you, because, you know, life on planet Earth is a really dangerous business. And to be a highly evolved species like ours that survived this long, you need lots of different layers of defense mechanisms. So all of that toolkit, gets thrown at trying to understand long COVID. And we and other people measure loads of stuff. So as I said, we've done it in our UK adult and child cohorts and in a Brazilian cohort. People all over the world have done similar things, and, you know, counted all kinds of stuff and tried to work out how it marks out people who have long COVID as being different from people who've made a full recovery from their COVID. And the much harder question, how any differences that you can spot, and you do spot differences, could help to explain the symptoms of long COVID that we all know so well, that, you know, a source of the trouble, in a way that could help us to, you know, intelligently design clinical trials and get to therapeutics. Okay, so lots of stuff gets measured. The stuff that I believe in my hands in our study as being different is that I think that if you look at the B cell compartment, the cells that are primarily there to make antibodies, for the people who've had COVID-19 who now have long COVID, their subsets of B cells are different, particularly some of the kind of early, what we call naive B cells. And part of the signal you see in that is not just counting the B cells themselves, it's the stuff they make. So obviously the main thing they make is antibodies, and antibodies come in many different flavors that immunology labs like measuring. And so the types of antibodies of immunoglobulins you can find in blood of people with long COVID, we think are different. And that's, if you like, a biomarker of long COVID. And the other biggie for me is that when I said how hard it is to link up immune differences that we measure to symptoms, one place you probably ought to look, if you use that metaphor of, if you lose a penny on a dark night, look under the lamppost, is at autoimmunity, because, you'd say, well, there's aspects of long COVID that look like a lot of things that we think of as being kind of autoimmune in nature, things like lupus and POTS and vasculitis. Perhaps there are some kind of commonalities or resonance there. Let's have a look if there's any new autoimmunity as a consequence of having long COVID. And I think there's really quite a strong signal there, yeah? So the first clue that you have, to me, some of the most convincing data that you could ever have, and we should always kind of shout this at naysayers who don't believe in long COVID or don't believe that COVID-19 does anything to you, which is that if you go to countries that have national electronic healthcare record data year by year, 2020 obviously is a very interesting year to look at, isn't it? Because most countries didn't have any vaccines on board yet. So, and they also were using, if they were lucky, PCR testing or antibody testing to know which part of their population had COVID and which part didn't. So there are quite clean data sets in many countries where you can carve up the country into had COVID versus didn't have COVID. And you can then follow all of those people, in most cases millions of people, year on year afterwards, and say, well, if you had COVID, how did that affect your risk thereafter of getting any autoimmune diagnosis? The whole shebang, type 1 diabetes and rheumatoid arthritis and vasculitis and IBD and, all the others. And the answer is, for almost any autoimmune disease that you care to look at, it raises your risk 2 to 3-fold. Yeah. So, and this isn't on sort of small sample sizes, this is on whole countries. So that's your first clue that once you've got that virus into your body, it's doing something to your immune repertoire that can trip you into autoimmunity. And then what people like us do is we look at every protein in the human body and say, do people who have long COVID make antibodies to those proteins? Because after all, that's what autoimmunity means, doesn't it? It means making antibodies or T cells to your own body, to your own proteins. And if they do have antibodies to those proteins, are there any patterns? Are there any clusters? And are they different from people who made a full recovery? And I think we think, and other groups think, that there's quite a sort of florid pattern of autoimmunity in people with long COVID. And some people have quite big clusters of different antibodies against different parts of their body. And that feels potentially useful and illuminating to me, because then you can begin to join up the dots and say, oh yes, so that might explain why that group of people have those symptoms. And sorry, I'll shut up a minute, but I just want to say one other thing about it, which is that the reason I like it is because I'm a very big kind of evangelist for immunology, and think we've done a really good job in post-war medicine. And one of the things I mean is that we've got a really good toolkit of therapeutics we've developed that make people better from stuff. So if you look at autoimmune diseases today compared to the start of my career, if you look at cancers today compared to the start of my career, immunology has really delivered. And we've got a load of goodies that we can give people where once you've made the diagnosis and you know the mechanism, you can make them better. So, treatment of irritable bowel disease, treatment of rheumatoid arthritis, and treatment of most cancers looks nothing like it looked 20 or 30 years ago. And the big difference has been immunology research paying off. And all the stuff that we used to write at the end of our grant applications in those days, if you give me the money, we'll develop a drug and it'll make people better, that we only half believed ourselves, kind of came true. So that's sort of advert for immunology to say, if you stick with us in this mechanism, there's hope out there.
So there'll be people who will just be having really mixed feelings about what you've just said. Like, this is amazing. We've got all this knowledge and we're finding out stuff, and we know that there's this risk and there are potential treatments that we can give people. And then they'll be, on the other hand, going, but anywhere I go, I'm coming up against a blank wall. No one's offering me anything. I'm not even getting through the door of the doctor's surgery. I've been discharged from clinic. And there's this real disconnect between the research world and treatment. And I think we'll circle back to that, Danny, but I'm just acknowledging that I think that there's, and I feel it as well, the frustration, and I'm sure you do as well, that you're doing all this amazing work, but it's not leading to anything meaningful for patients yet.
Yeah, everything you just said, exactly.
Yeah.
And I promise you, all the people who are currently bashing their heads against their laptop saying, what the hell is he talking about? I haven't seen any of these benefits. I couldn't empathize more. But please, stick with me and keep up the optimism. One of the expressions I use quite a lot at the moment is tipping point, because I do think we are at a tipping point where we've gone from standing start to quite a lot of knowledge, and we probably know as much about long COVID in the last 4 or 5 years as many other immunological diseases in the last 100 years. So we are kind of catching up quite fast. And sure, yeah. The trick now is to convert all that research into something that actually helps somebody get better. All I'm saying to that is, honestly, immunologists do have a track record for getting people better. So bear with us, and I think we're nearly getting there.
Yeah, and so you were one of the organisers of the Santa Fe conference, which was a Keystone Symposium. Just tell us what that means and what did that involve? What did that look like?
Yeah, sure. So if you're in the kind of biomedical research world, Keystone meetings are a kind of a big deal in the calendar, because obviously we all go to conferences all the time. And the Keystone meetings are something a bit special. They're very, I suppose they're quite elitist, and they're the place where you have these amazing venues and the world leaders get together. And the sense is that you're getting the sort of biggest brains on the topic and knocking heads together to try and make a difference. Okay. So we had a group of us who were asked by Keystone to organise this long COVID meeting this time. And so I should say who they all are. They're Hannah Davis, who a lot of people will know as, one of the key sort of patient activists, and Avindra Nath, who's at NIH in Bethesda, who's been the head of the ME program at NIH, and myself, and Akiko Iwasaki from Yale, who's been absolutely one of the biggest, most prolific figures in long COVID research and long COVID clinical trials. And so we got together, God knows, about 9 months before the meeting, and I think really were on a mission that, sometimes the Keystone meetings can be a lot of fun, where you do a lot of skiing and drink a lot of alcohol. And we were determined that we weren't going to have one of those meetings at all. And nobody was going to do any, well, they couldn't do any skiing because it was in the New Mexico desert. But, we weren't going to kind of have wild parties or anything. We were just going to really knock heads together and try and take this really, really seriously, and try and have this be a sort of landmark meeting where we made an increment and made a difference. And I think everybody who left thought we really, really had. So
all the absolute rock stars of long COVID research were there. You did an incredible job getting the right bums on seats, that's for sure. And what do you think? There was so much. When you look at when I looked at the program, I was like, wow, there is so much. It was almost overwhelming to look at the program, to go and kind of go, wow, there is actually huge amounts of research going on all over the world, right? Yeah. And can you tell us the kind of the big themes that were discussed in terms of okay, so we've gone from knowing nothing 5 years ago, and now we've learned a lot. What are the big themes that you think will be taken forwards in terms of the areas of focus for research and where we might see therapeutics like drugs and medicines developing?
There's probably a handful to talk about that are really, really important to get across. Let me start with one of the biggest and most obvious that everybody will know about. That there's been so many sort of branches of long COVID research, haven't there? But almost one of the most sort of predominant that's come to the fore overwhelmingly has been this notion that SARS-CoV-2 can do something that we didn't think coronaviruses could do, but we should have known, which is that long after you've cleared your initial acute infection, it can linger in the body as a virus reservoir. And we knew about that for other viruses, and we didn't think about it so much for coronaviruses, partly because we were very obsessed by things like the common cold and weren't really thinking broadly enough. But if we had been thinking, we'd have been on the lookout for it, because obviously a key disease in cats caused by coronaviruses is sort of persistent feline coronavirus. So we should have been on the lookout for it, and we didn't think that the virus could behave that way, but it so clearly can. And if you're trying to join up the dots and think, well, how could this coronavirus still be causing disease in me 3 or 4 or 5 years later? If you've still got the virus on board, driving a sort of ongoing persistent inflammatory response, that might be quite a good way of explaining long COVID. And I thought there was so much data presented on this that was really, really compelling. And if you put it all together, and what I like about it is it's not one test, it's different labs asking the question in different ways and coming up with a similar answer. So, there have been glimmers around for a long time, haven't there? From and one of the most obvious that I really liked was the sort of the olden days stuff from a few years ago, saying that if you take people who happen to have been referred by their GP to a gastroenterology clinic and have to have a gut biopsy, to confirm or refute a diagnosis, and then you correlate the findings in the gut biopsy with their long COVID symptoms, the people who reported the long COVID symptoms were the same people where you could see bits of coronavirus in their gut. So I always liked that data, and that seemed very compelling, and there's lots of data like that. And that sometimes, but not terribly well, ties up with the sort of microbiota studies where you PCR virus out of people's poo and shows that correlates with long COVID, which is a slightly less good way of doing it. And probably my sort of favorite gold standard way of doing it comes from Mike Peluso's lab in San Francisco, where they do PET, positron emission tomography that you can do in any hospital in the UK, but they don't do the normal one. They do a really fancy Californian one where they've got a PET tracer that lights up activated T cells, which unfortunately we haven't got our act together to do in this country, and we need to. And if you do that on people with long COVID compared to people who don't, you see these really lit up T cells in the gut where the virus is in long COVID. So if
if I may interrupt you for a sec, because I think that's I just want to pause on that, because I think there's lots of people who obviously see doctors, they go to clinics, they have lots of tests, standard NHS sort of tests done, standard scans, and it doesn't show anything, and they get told, you're fine, discharge, go home and think yourself better. And I think that's really important to highlight, that there are advanced imaging techniques, advanced blood tests, there's advanced assays and things that can be done, but they're all confined to the research world at the moment that do show these changes and do kind of start to validate some of the symptoms people are experiencing.
Yeah, I mean, it's exactly that. So I'm so aware of the frustration of people I meet and you meet with long COVID who've been sent away saying, well, we did the tests, they were all normal, get lost. And and I know that the answer to that is, well, sorry, you did the wrong tests. Because they're great. You still with me? You okay? Yeah. There are great tests out there if we had the will to roll them out and use them, and it's up to us to to pressurize and say, well, actually, perhaps your old tests aren't good enough for what I need. Perhaps I need the NHS to be using new tests that can actually tell me what's wrong with me and do something about it. And there are many examples of that. So
yeah, I think that was just a sidetrack slightly, but I thought it was a really important point, and one that it will be important to the audience. Going back to the viral fragments and the viral resistance, and I think there's been another whole session, another live stream if people are interested in this, where Amy Proell and I chatted about this and really broke it down, because there's a difference between the virus is there, it's live, it's replicating, it's gonna respond to antivirals, versus the chunks and fragments of the virus that are just acting as these little kind of foreign invaders, if you like, and then your immune system responds to those little pieces of the virus. And can you tell us a little bit about the work that's been done on antivirals and what that's telling us, and what the discussions at the conference were around that?
Yeah. Obviously antivirals have been kind of at the front of the pack in terms of big clinical trials for very good reasons. And at the conference, there were four different reports from some of the participants there, reporting failed trials of Paxlovid, which people will be very familiar with. So you might think that if all of the Paxlovid trials had failed, people would be very downhearted that was a kind of false avenue of investigation, and B, that maybe it kind of refuted or disproved the idea that there's a virus reservoir somewhere, and if we could get rid of the virus reservoir, we'd make people better. And I think neither of those views were particularly prevalent. The way I describe it is everybody who presented that data said that this was just a kind of first springboard to the next generation of trials, where if we could do them learning from the first generation more insightfully, then we could get cleaner answers and get the answer, because the answer does lurk somewhere in there. And part of it relates to the question you asked about whether it's real, bona fide, treatable virus or some sort of viral particle. And the other thing I think it sort of signals is one of my other kind of key themes from the meeting is stratification. That anybody who knows or cares or thinks about long COVID knows that it's a really heterogeneous umbrella term for lots of different people with lots of kind of overlapping but different things wrong with them. And one area where I was frustrated at the meeting, despite all our efforts to get clever people in the room and knock their heads together, was that I didn't feel like as a field we'd made intelligent enough progress on stratification. And I think that's really, really holding us back on everything, on treatment and therapeutics and diagnostics, and having clean clinical trials that can give us clean, meaningful answers. Because, I think most people are onto the idea that if you think about people with diabetes, there's a huge world of difference between the child who has type 1 autoimmune diabetes and the kind of older, often higher BMI adult who has type 2 diabetes. And they're kind of not the same disease. And if you try to treat them the same way, you wouldn't really get very far. And I think we're in the same. Yeah, I think we're in the same mess at the moment with long COVID. We're trying to treat everybody in the same huge basket, and not giving enough brain wattage to how to separate them out. And
100%.
Yeah, but
yeah, 100%.
Yeah, but when
and I think it's a massive barrier, isn't it? Because if you just lump everyone, it's like saying let's treat cancer with this drug when there's 100 different types of cancers, and you actually need, now we're down to the individual receptor on the individual cancer, and that's how precise medicine has got in the cancer field. And yet we've got this blunderbuss kind of let's lump everyone together regardless of how they're presenting and their symptoms. So I mean that's we always have to sort that out before we can get the good treatment trials, don't we? But I want to just go back because I think that's really encouraging that we haven't just binned antivirals because the first few trials have been negative. And we have to remember that they only treated patients for maybe 1 or 2 weeks, and that's probably not long enough, and et cetera. We've got the Mark Fahy's trial that he's done in the UK that's going to hopefully be reporting soon, the IV remdesivir trial. And I know there are some other antiviral trials going on in the States, which is helpful. I get a lot of questions, Danny, about spike protein, and I always struggle to answer them because I think if I measured the entire population of the world, lots of people would have measurable spike protein in their blood, but not everybody is sick. And of course, there are some monoclonal antibodies that will target and go and bind, spike. What's the general thoughts in that kind of global field about the relevance of measuring a spike protein and seeing if it's floating around?
Sure. So but I mean, there's kind of two questions lurking in your question, isn't it? One is it kind of meaningful or helpful or illuminating to measure spike in blood? And the other is kind of, could there be any spike lurking around in this virus reservoir, and therefore could it be useful to treat people with monoclonal antibodies against spike? And that's another way of getting rid of, well, of persistent infection and of long COVID. So I'm not, I don't want to kind of rubbish anybody else's research, but my sense of looking at quite a lot of people's data and our own is that I don't think there's a really convincing signal that, even if you use quite fancy, quite sensitive techniques like Simoa, which is like the most fancy, high sensitivity way that you can measure that kind of protein in blood, I don't think you see a really convincing signal that it's there in the majority of people and differential between long COVID and recovered from COVID. And the same goes for nucleic acid as well in our hands, in a really kind of comprehensive study that we did. That doesn't mean that I rubbish the idea that it could be useful to do clinical trials, or more clinical trials than have been done up to now, on neutralizing monoclonal antibodies that bind to spike and could get rid of any residual virus like that. And again, we've seen a few small trials reported, haven't we? One of which looked very optimistic, and one less so. And I would just love to see more and more and more of that data, or even do some ourselves if we can, and bottom that out, because I think there's a good chance that might work.
Yeah, and I think there are kind of, I mean, we can't really get hold of monoclonals in the UK, at all, really. Even if you're trying to buy them privately or what have you, it's very, very difficult to, and the new one, Pavabart, isn't even licensed in the UK or the US. It's only got a license in Europe, so it's very difficult. And my understanding is the drug companies are not that interested in repurposing these drugs for long COVID. That's not what they're licensed for. They're generally licensed for immunosuppressed folk. So was there a sense that there's going to be a drive for monoclonal trials when you're at the Keystone, or is it a bit dead in the water? So I don't want to sort of
it's absolutely alive and swimming for its life. But as you said, maybe harder in the UK. And I've certainly been involved in some discussions with some of the companies who make some of the monoclonals, which, as you say, was like a kind of failed speed dating exercise, and they'd move on the table quite rapidly.
Yes, and I think this is part of the challenge that, if we look on a global scale, how could we get all of those people to come together and go, well, actually, we've got, millions of people around the world. We've got these drugs. We need people to come together to want to come up with a solution, and that's really got to be driven by governments and policymakers. But anyway, I guess
struggle. Sorry, me connecting up my laptop that was about to die, but I promise you.
Oh, right, that would be a disaster, so we don't want that to happen. Okay, so we were kind of talking about the big themes that came out of the conference. So viral persistence, viral antivirals, targeting, that was one of them. We're suddenly short of time because I told you this would go really quickly, as I do want to bring Helen back on in about 5 minutes. Was there any other big sort of headline kind of going, this is looking promising, that's looking promising?
Yeah, I just want to say one or two sentences on the word biomarkers. So, what do I mean by biomarkers? I just mean that everybody who does a disease study, there is a holy grail, isn't there, to say, I've, measured the blood of 500 people with long COVID and 500 people who made a full recovery, and I've got my signature in blood of some proteins or antibodies or whatever it is I can measure that defines people with long COVID. And that sounds kind of prosaic, but the reason it's a holy grail is that, it opens up so many doors, doesn't it? Because first of all, it gives you a diagnostic test that your GP can order. They can tick the box on the form and send it off to the hospital lab. and therefore you can define who has long COVID, who hasn't. They've got validation, they've got recognition, they've got entry into a clinical pathway. They've also got entry, hopefully, into clinical trials where you can say, these are my defined patient group, they're different to my defined control group. And when that biomarker has gone away, I'll be able to say they're better. So, it opens up all those doors. And anybody who's been following this field knows how frustrating it's been that amongst those thousands of long COVID publications are several hundred that claim to define a biomarker. This was different, or that was different, or that was measurable, or the other thing was measurable. And probably the sort of loudest rant I made at the meeting was where I sort of forced people to grow up and stop just sort of publishing things for the sake of their CV and do what we do in other fields in the past for this, which is agree to have an international workshop where we all kind of rank our biomarkers and exchange our patient sera and say we're damn well going to validate which are the ones where we have consensus, where we all agree. And when we found the best ones, we'll call those the biomarkers and no others. And it has to be a test that's doable, where I can get the same answer, whether I'm in Halifax or Honolulu. And that'll be the answer, and that'll open up all those doors. And I think we've now got agreement to actually do that.
Yeah, sorry, I lost you just for a second there. And I don't know if it's my signal or yours. But, that's amazing if we're making progress towards that, because that really has been one of the biggest problems. Every day I wake up to a new paper that's claimed they've got the latest biomarker and the latest answer to the diagnosis. If we could get the global community to come together and work together on that would be incredible. Excuse me. So, was there anything else, Danny, other than the virals, the biomarkers? Is there any other really sort of landmark studies, trials that we should be excited about just in the last few minutes before I bring Helen back?
I just think, when I use the word tipping point, I think there's a lot of trials either in progress at the moment or being kicked off in the next year. So, it's frustrating, but it's very much a matter of I just if we okay?
Yeah, we're sorry. I think I don't know if it's just me. I'm going to bring Helen back on stage. She might be able to tell us if it's my signal or did you lose Danny there as well just for a moment?
Yeah, unfortunately, we did miss you a little bit there, Danny.
All I was saying is, that I really, I know I keep saying it, but I honestly think there's hope, and if we can look at a tiny bit more immunology and look at all of these trials that are coming online in the next year or so, I really do think we'll get there.
Yeah, honestly. Amazing.
Here's hoping.
So we're just still having a few issues with you going in and out, but we'll do our best to carry on. So Helen's been keeping an eye on the chat, and I'll bring you in just a sec. I think it's really important to remember that Danny is not a clinician, and there's probably a lot of clinical questions that are being asked. So I am a clinician, and I'll do my best to sort of chip in if needs be. But yeah, Helen, what are the sort of things that you think are coming through in the chat, and have you got any questions for Danny?
Yeah, I mean, it's so tricky, isn't it? Because it's such a broad topic. And like you say, to try and treat everybody with the same thing is nonsensical, because it's not the same thing for everybody. But some people have the same elements as other people. Some people have the whole bingo card, and some people only have one or two things. So it is very tricky, and it's tricky with the interview as well, of course, to cover everything for everybody. But thank you for managing to do that in the areas of both of your work, to be honest, because it is such a huge spectrum and it's impressive. So thank you for fighting our corner in the way that you are, both of you. Yeah, there is a lot of clinical questions as well. Yeah, I mean, the title of our channel is What You Can Do. And so I don't know if Danny, you've frozen. I'm not sure if you can you hear us okay? Is it just freezing coming our way?
If I dance around, I look less frozen.
Fair. So a question I often ask is, what you can do. So if you had some advice for patients, bearing in mind the broad umbrella of who we all are, what are some key things that you think it's crucial that you do this? Like maybe, I don't know, keeping a diary, and who would you give that diary to, or things like that. Is there any kind of tips or advice that you would think are crucial? So go back to basics.
Yeah, I mean, so well, obviously, the basic was the stratification and the real kind of granularity in this complex disease, and I think that as we get to the stage where hopefully we do have some better therapeutics and some better ideas of which way to go with therapeutics, the better you can understand what's going on with you and the better you can describe it, the better off we're all going to be. Because when I describe my frustration with this heterogeneity and stratification, I know there are some people out there who want to be able to sift all the long COVID patients into very simple pots and say, oh, you've got to go to the psychiatric team and you've got to go to the gastro team. But anybody who's really ever dealt with that symptom spectrum knows that's a bit bonkers, because you could array 40 different symptoms and everybody would look like a scattergun of having quite a lot of PEM and some brain fog and some of this and some of that, and they'd all look kind of overlapping but different. So we're not really going to kind of sift people into two pots. We're going to so I think just understanding your symptoms and the kind of granularity.
I think we've lost you again there, Danny. Shall I come in? Because I can probably have a crack at some of the clinical questions while waiting for Danny to come back to it. Yeah, and I think there's going to be a delay now, so there might be him sort of popping back in and while we're trying to chat. But yeah, Danny, I think Danny, we're losing you. We're having a bit of a nightmare with the sound. So I'm just going to have a crack at answering some of these clinical questions. I don't know if you want to jump off and maybe jump back on and see if we can get a better connection just for the last 15 minutes. That would be great, Danny.
Well, I was just going to say, if you make sure nothing else is on, no other windows are open, things like that. I don't know if you've already done that.
Shut down. Yeah. But feel free to jump off and jump back on. So yeah, I mean, I think basic things it's really difficult, Helen, because I spend at least an hour with every patient doing a very detailed history and then bespoking that, even the basic advice. But I think the key thing is rest and pacing, okay, in terms of practical. I mean, I know that sounds like oh god, she's talking about resting and pacing. We know how to do that at this stage, but I can't emphasize just how important that is on top of trying to do absolutely anything else. And pacing isn't just about doing less, it's doing differently. So you can be pacing and have a shower, but actually that shower, if you're not doing it in the right way, could really trigger symptoms and fatigue. So I think there's a lot to pacing. I've done several sessions on this on the channel. So watch the Todd Davenport one, watch the one with Robin McNellis. There's a lot to even just pacing. I think getting a diagnosis of POTS, if you've got POTS, because that's completely treatable. We can, when I say treatable, we can get symptoms under control, and actually just really simple treatments can actually sometimes make a massive difference. Just slowing somebody's heart rate down, giving them salt, giving them fluid, that can make huge amounts of difference. I think looking at diet, nutrition, like, are you having anything that's triggering your immune system that you could be taking in orally? Like, there's been a lot of questions about mast cell activation in the chat. If you're histamine intolerant and you're having loads of high histamine foods, which are really healthy, by the way, it's tomatoes and avocados and lots of citrus fruits. Yes, all of these things are high in histamine. If that could be, if you're taking in something that's just constantly irritating your immune system and creating inflammation, it's looking at that. It's making sure you sort out your sleep, getting decent sleep, getting into good routines. Sometimes melatonin and things like that can help. So there's lots. I think long COVID recovery is very much a jigsaw puzzle. There's lots and lots and lots of different things. I often say to patients, this is like a full-time job recovering, because you have to almost look at every single piece, one of those little pieces of the jigsaw, and then put it all together. And then, of course, medications for symptom support, treating inflammation, all of that plays a role. And of course, I can sense the frustration in the chat that most people don't have access to those, and you can only access a lot of those treatments in private clinics, and people like me who want to do it in the NHS can't, because there's no the commission services don't allow you to prescribe. So it's a bit of a desperate situation where even if you want to treat people, you sort of can't, because there's no guidelines or pathways. So we end up in that, but there is a lot of stuff people can do by themselves. Danny, I know this isn't really your bag, giving people practical clinical advice, but is there anything you'd like to add to that?
I try and steer clear of it if I can.
Yeah, that's fine. Okay, so let's get back to the immunology questions, because they are sort of coming in. There is one that there's a few questions that have come in about commercially available tests. So some people are getting tests that show they've got spike protein in the blood, and there is one test that's done in Germany where they'll tell you that there's spike protein in the exosomes and in the plasma and in the white cells, and and then there's another company that's doing tests to look at COVID antibodies and saying if they're low, and they're looking across lots of different strains of antibodies, and if they're low, that's sort of an immune deficiency, and that's it's a problem with producing antibodies, that's the issue. Or maybe the antibody levels are really high and that's the issue. And these are all sort of tests that have not been tested on hundreds and thousands of patients. And individuals are taking these tests and going, oh, look, this is mine. They bring them to me and go, this is my individual result. What should I do about this? What treatment should I have? Have you got a view on kind of that generally?
Yeah, I mean, let me try and present some kind of measured view, hopefully without sounding too dismissive or snooty or anything. So my day job has been doing really fancy tests on, well, in fact, thousands of people from different parts of the world in COVID-19 and long COVID for the last 5 years. So I've looked at all these tests in my own hands in really big data sets. And there's variability, yeah? So whether you're people who have long COVID or don't have long COVID, there are people out there who have high antibodies to spike and low antibodies to spike, and high T cells and low T cells, and high cytokines and low cytokines. And it's been really hard to define a sort of signature of long COVID for practically any of those things. So, I mean, I'd only single out a few observations that people email me about. There are people who email me a lot and say, I've ordered a test and I've got a really low CD4 count. Does that mean that long COVID is a bit like AIDS and I've got something AIDS-like? And, please let me say, sure, there can be individual differences, and some of us are high for some things and low for other things. But this really isn't an AIDS-like disease, and most people with long COVID really aren't substantially immunosuppressed. So that's a kind of long introduction to saying all of these tests, the commercial tests for individual immunological assays, if you're interested in some kind of self-insight, by all means do them, but I don't think they can't be overinterpreted. They can't be interpreted in any useful way for a single individual person. They can't inform any particular treatment. And they're just, a way of finding out interesting information about yourself. I don't think they illuminate the diagnosis or the treatment at the moment.
Yeah. And I think I sometimes use the example of, if I invented a blood pressure cuff and I measured your blood pressure at one point in time, and then you came to me and you said, what does this mean? And I said, well, I don't know, because I don't know what your blood pressure is like every day. And if we did it over, several months, and, so you go to the GP with a one-off blood pressure reading, they're never going to put you on high blood pressure tablets, because you've got to demonstrate that that's repeated issue. And then also I might say, well, actually, I don't want to measure it. I want to measure blood pressure of the whole population, so I understand what normal looks like. And then I can tell whether your results are outside of the norm. So I guess what I'm saying is there's a way of validating these tests to look at what's normal, is it repeatable, is measuring something at one point in time giving you the whole picture. And then once you've got all of that information, you could take a test and say, well, this is how we do the test, this is how we interpret it, and these are the normal levels. And then you can take that test and maybe use it in a study to say, well, let's now see if this test tells us whether the response to drug A or B is working or not. So that's the process of validation. And I think the thing that I know there's a lot of stuff out there that has only been validated in small numbers and not large numbers because of the lack of research. And I find it quite difficult to advise people looking at the results of a one-off test at a single point in time. I don't know what your thoughts are.
Yeah, no, it's exactly that. And the blood pressure and hypertension metaphor is useful, isn't it? Because when your GP gives you advice on that, it's kind of quite, even if you haven't had 24-hour monitoring, it's kind of well-informed advice because it's based on a data set of not millions but hundreds of millions of data points. If I've measured my antibodies to spike or my serum IL-6, that isn't based on that data set at all. So you can't interpret my individual data.
Yeah, and I think this is, and I see it from the other side. So I see the patients who are just desperate to measure anything to validate that there's something going on. There is often tests that give very subtle signs that there's something going on with the immune system. So a lot of my patients will have a slightly low neutrophil count or a slightly low lymphocyte count, or they'll have a slightly low CD4 T cell count, but certainly nothing that an immunology clinic is going to get interested in because they don't meet the threshold for being immunosuppressed, but there's a signal that something's not right, maybe the immune system's not functioning properly, and they're stuck in this kind of bit where no one will do anything because we don't really know what it means. And do you see those immune system abnormalities, Dan? You're not saying that if somebody's not completely immunosuppressed, there's nothing wrong. I guess what we're saying is we just don't know how to. The research hasn't been done yet to allow us to actually treat effectively.
I think it's that, even if you go beyond, the really sort of fancy experimental stuff that labs like ours do, if you're lucky enough to be near a really good hospital that has a really fancy clinical immunology department, many of their tests have only been in the NHS for 5 or 10 years. And they're very, very much at the sort of outer edges of things that can meaningfully be interpreted at a sort of population level and maybe inform treatment, but barely so. And so the kind of things we're talking about are even beyond that.
Yeah, there's a few questions about vaccination as well. Helen, I don't know if you were keeping an eye on those. I did spot them coming in. I'm just trying to, so many comments now. I'm trying to find them.
While you do that, just to finish this off before we move on, I just wondered if you could talk a bit about the way that levels do change. So people have overcompensation and dysregulation of various things. It could be microclots and then bleeding disorders, or it could be heat regulation. If it's hot, you get very cold, and vice versa. And is there any commonality between those things and the body kind of over compensating for what should be happening? Is that something that you know much about? Because there doesn't seem to be hardly anything out there about it.
The only answer I'd give to that is just that, as everybody, bodies are really complex homeostatic symptoms where for everything that gets turned on, there is something to turn it off again. And yes, sometimes they overshoot. We've had the Nobel Prize for Medicine 2 days ago for 3 people who looked at precisely that, about how the immune response is actually turned off by regulatory T cells. So, as part of the analysis we've all got to do is looking at, radiation is going up and down and going up and down incorrectly, which is just a different way of saying disease, isn't it?
Yeah. So there's quite a few. There is a few questions that have come through about vaccines, and I think the themes are generally like, what do you think about having a vaccine if you've got long COVID? Is Novavax better than an mRNA vaccine? And then there's a couple of questions about post-vaccination syndrome, which I think is really important to acknowledge and recognize. And I'm wondering whether at the Keystone Symposium there was much discussion about this topic. And so there's 3 questions in there, Danny, and I appreciate we haven't got very long left. So any thoughts?
I can pick them off, I think, fairly easily and uncontroversially. You know, people will be very familiar by now with the data sets on the effect of vaccination on long COVID, which is, you know, people had hope of a reservoir of virus. And I think in the meta-analyses, you know, the combined analyses, it's kind of maybe a third of people with long COVID noticed improvement after vaccination, a third noticed no difference, and a third thought they. I think about it is that if you look at really big data sets, like the famous data sets from America, from Ziad al-Ali, the risk of
we're just losing you again, Danny. Sorry.
COVID infection. I'm not quite sure what to
yeah,
do.
We just lost it. So I think
you're back.
We had the rule of thirds. About a third of people get better, a third of people no change, about a third.
So the next stage in that is that any repeat infection with COVID carries a new risk of long COVID and a risk of worse long COVID. So just avoid it. And the best way to avoid it, I think, is to get vaccinated. I've got no preference between mRNA vaccines and Novavax, because I think they're both amazing and brilliant, and I'm a big fan of both, and they both induce absolutely brilliant immune responses.
Yeah, and
so
I
I'm
think
a big fan of both.
It's one of those that if you are one of those individuals who's been unlucky enough to develop a post-vaccination syndrome, and and I have seen some patients where that has been absolutely devastating, and it's very difficult to prove. People don't take it seriously, and I think they are the unluckiest of all because they had it in good faith and they developed very severe illness. And there is some interesting work going on by Petrino and various people in the States where they're looking at the mechanisms. And my view is we should just really try and study it and try and understand it and make the vaccines better, not pretend it's not happening. But given the sort of billions of doses that have been done, overall there is undoubtedly a benefit and a protection against long COVID. But it's that, again, going back to that individual risk, it's very, very difficult. It's very difficult for individuals who are poorly with long COVID and they're being advised to have a vaccine. They're a bit scared of having a vaccine because they're worried about having an immune response to something that, so I think it's difficult for a lot of patients, but it's reassuring to hear that overarchingly, the vaccines are protective.
Yeah, no, I think in the sort of big scheme of things, you're so much better off with the vaccine than without. At the Keystone meeting, this syndrome, which clearly is a thing.
We're losing you again, Danny. Sorry.
Who are post-vaccination, so it's a thing.
Yeah. So okay. So I think the gist of that was, it's absolutely being recognised. No one's pretending it's not happening. We need to study it.
Okay.
Just in the last minute, there's a lot of questions about NHS and rolling things out a lot in the NHS, and why, given everything we've said, is there still nothing? Why do immunology clinics still ignore MCAS? Why? Yeah. And I think, and I can sense the frustration again in the questions that are being asked, because this is coming from people's experiences. And I think it's very, very difficult in the NHS to do anything outside of recognised guidelines, proven treatments. There's very, very rigorous processes that things have to go to be approved by NICE, to be rolled out. It takes a lot of time. And actually, if you want to do something different outside of the box and treat patients outside of usual processes, you're actually not allowed to. So, doctors will get disciplined for doing things like that. So if the National Society for Dermatology or Allergy or Immunology does not recognize MCAS, does not train its doctors in MCAS, does not have guidelines for MCAS, doesn't have licensed treatments for MCAS, the NHS cannot deliver those treatments. So, and there's no education and training about it. So people aren't deliberately doing the wrong thing or not doing the right thing. The systems that you have to work in, if you work within the NHS, are very, very stringent. And because drugs are paid for from the public pocket, there's very strict rules about what you can and can't prescribe. And while there is some leniency, it has to be done through certain processes. And so until we sort out the research issue, I think it's going to be very, very difficult for these sorts of things to become mainstream in the NHS. So Danny, back on that point, what's the call to arms? What do we need to do to drive this forward for the community?
I'm afraid there is a call to arms. I know that people with long COVID are already busy and exhausted, but at the end of the day, we've got 2 million people in this country, 400 million people in the world, a cost to global economies of $2 trillion a year. Politicians, even if they didn't have empathy or a heart, ought to be kind of economically convinced that it's useful to have this many people out of the economy, and so why not support people with long COVID, support more research and accelerate treatments so that everybody can get back to work and get back to their lives and get better. I'm afraid people do need to shout at their MPs about this.
Yeah, I mean, I think as someone, and Danny, I know you've done a lot of this as well, advocating in Westminster and talking to MPs about this issue. If it's not visible and it's not on every MP's table, it's not something that can be raised at that level. It's not something that can be prioritized. The more we can get people, even though you might feel like you're screaming into the wind and it's pointless and you're not going to do it, if everyone did it, if all 2 million people in the UK did it, that would have an impact. So I think it's thinking about how we galvanize. And of course, people shouldn't have to advocate for themselves. But ultimately,
if I could interject, if that's okay, I know we're running out of time. We've run out of time. But on that note, if I could do a call to action for people, POTS UK has actually got a call to action right now. It takes less than 1 minute. Obviously, there's lots of different types of long COVID, and one big subgroup is POTS. And if you can help one subgroup, you can help all the subgroups, because we're all kind of, here for each other. But if you go onto the website, the Westminster Hall debate on POTS is due to be Tuesday, the 14th of October at 11 a.m. Now, we want as many MPs as possible to attend. So if you go onto the website, it's just a basic form, fill out your name, your email address and your postcode. It will tell you who your local MP is. It will send the letter on your behalf, and so your MP will be emailed and encouraged to go on the 14th, at 11 a.m. And just, if we can raise awareness as much as possible, and if they get inundated with emails, exactly. I have heard from MPs previously that they struggle to represent long COVID because they haven't heard from people in their constituency. Well, that's going to be tricky because a lot of us are not well enough to be doing that. But if we can do that when we're able to, and if we can ask family members or friends and anyone we can, and get as many emails out there as possible, it really truly does help, and it does, help all of us. So yeah, POTS UK, we'll put the link in the thing, because although I'm here supporting long COVID, support the charity, there's lots of charities all fighting for the same thing. And so, yeah, thank you in advance for that.
Absolutely. Well, I feel like we were just really getting warmed up there, and now we've had to, we're going to have to close the session. I'm very sorry about all the sound issues, and it was a shame because, this happens unfortunately when we're live streaming sometimes, but we did catch the majority of what you said, Danny. So thank you so much. And I think it's good to end on that call to arms. It's also good to end on a positive that, this isn't forgotten. There are people from all over the world coming together. There are people like Danny and colleagues from lots of different countries really fighting hard. There's lots of us advocating at very high level to change things. And, if everyone can, if we can just get as many people behind us as possible, just to do it, even if it's just one tiny little thing, it makes a big difference. Lots of information on the Long COVID support website as well. And thank you to everyone watching, because you've all been brilliant and brilliant questions. Sorry we didn't get through them all. So we'll be back in a few weeks. My next guest is Dr. Harriet Carroll, and we're going to be talking about POTS and thirst and water, and it's going to be it's going to be a really interesting discussion. Danny, any final bits from you?
No, just remember we're at a tipping point and we're getting there.
Brilliant. Thank you so much, and I will see you next time.
Thanks, everyone. Thank you, Danny.